Good evening, children. In the lesson that we started, absorption by roots, the processes involved, till now we have studied about the general characteristics of roots, the functions, and what are the main characteristics for absorbing water. Along with that, we have done the phenomenon involved in absorbing water and minerals for transportation and upward conduction. We have discussed 5 main important processes. For example, number 1, imbibition. The second was diffusion. Third was osmosis. We studied endosmosis and exosmosis. Then a very important table I had given you to learn it by heart, and this was the examples of different kind of membranes, that is impermeable, selectively or semi-permeable membrane, and then freely permeable membranes. We moved on further last time with active transport, the process of turgidity and flaccidity, that is plasmolysis. So these were up till here the 5 phenomena, children, which I said they are important, from imbibition being number 1. Then this was 2, 3, 4 and 5, right? Along with this, we move further with the explanations of plasmolysis, flaccidity, deplasmolysis, and different kinds of pressure which are operant: turgor pressure, wall pressure, osmotic pressure, root pressure. And then we had finished our class with active and passive transport with this diagram that I had explained about the structure of single fully grown root hair. Today we will move with our new topic, tonicity and 3 types of solutions. Tonicity means relative means comparative concentration of the solutions that determine the direction and extent of diffusion, right? And this is what we call as tonicity. 3 types of solutions. So that means if you have 2 solutions in contact, then tonicity is the relative concentration. That means which is highly concentrated or which is lesser in concentration, and accordingly, keeping this chapter, what should happen, right? So we have many hypothetical questions to come. First, let us study the 3 types of solutions. The first is isotonic. The term iso means same, right? This term isotonic means same. So I've written over here, and tonus means tension, concentration. The relative concentration of water molecules and solute on either side of cell membrane is the same. You'll have to understand this with an example. Suppose this is a plant cell which is immersed in an isotonic solution. That means the amount, or let's say the relative amount, of both water inside and outside is same, and not only just water but also solutes which are in the water that are dissolved or in the cell, they will be same. So in such a case, what will happen? Which direction should water move? Should it move inside or should it move outside from the cell? Nothing will happen. In this case, there will be no osmosis, and the cell shape and size remains unchanged, right? Once again, before we move to the other, in this case, as I said, isotonic solution, when we have equal amount of water and solutes on the outer side of the cell and the inner side of the cell also, there will be no osmosis and the cell shape and size remains unchanged. Now coming to the second solution, right? Let's say hypotonic. Hypo means less or lower, and tonic means the strength or concentration. So the solution outside the cell has a lower solute concentration. Solute means the dissolved substance or the solid, right? So the lower concentration of solute outside than the fluids inside. Let's see, we'll see this by an example, right? Suppose this is a hypotonic solution. Hypotonic means the solution outside has lesser solute than the fluids inside, right? So in this case, what will happen? In this case, water enters the cell. Remember children, anywhere where you have hypothetical questions, you have to always remember that where is water more or less. Hypotonic means the water is more outside than the inside, right? Because the strength is hypotonic and therefore the water starts entering due to endosmosis. Alright, so this is the case where you have to understand that due to lesser solute concentration than the fluids inside the cell, endosmosis begins. In the next category where we have hypertonic means a higher concentration, right? What will happen? If the solution outside the cell has higher solute concentration than the fluids inside, exosmosis will occur. For example, if we see this diagram here, hypertonic solution, suppose it is a concentrated sugar solution or a concentrated salt solution and the cell is immersed into it. So in this case, comparatively, water inside the cell sap will be higher than the water outside. Why? Because this is already concentrated. More solute like sugar or salt and less water. And remember children, osmosis or the movement has all the ways where it's more water and less water. So water starts moving out through the semi-permeable membrane and this is causing the cell shrinking in size and the cell loses shape. Alright, so here just see this leads to exosmosis. Alright, so that is what causes the cell shrinkage. And here in hypotonic, the cell slightly enlarges or there is a chance that it can burst also if it is placed in hypotonic solution. So I hope this is clear children. We've started our class today with tonicity and the three types of solutions: isotonic, hypotonic and hypertonic. Moving further, this diagram we have already explained. There are one or two technical terms that we have to study. Semi-permeable membrane, though I have already given you this table, but by definition let us see what does it mean. This table I have given you, semi-permeable, so many examples we did: cellophane paper, egg membrane, animal bladder, whisking bag, goat's bladder, pig's bladder, etc. But by definition, what does it mean? The definition says it is the one which allows passage of molecules selectively. That is, it allows a solvent, that is water molecules, to pass through but prevents the passage of solute, that is sugar or salt molecules in solution. So that is the definition of the semi-permeable membrane. Now in your ICSE questions children, there are many times questions on reasonings and you are given, like for example, why is salt added to pickles, or let's say excessive fertilizers damage the plants, why? Alright, or we can kill the weeds in the plants by sprinkling excessive salts, etc. Now these are practical examples of plasmolysis in practice. If addition of salt to pickle is there or excessive salt is added to meat, in both these cases what happens, the attack of bacteria is avoided because this bacteria is killed if it attacks these pickles due to plasmolysis. Plasmolysis means, you understand that the previous term or the previous discussion we have discussed this. I'll just show you here. Plasmolysis is this state. If the bacterial cell attacks the jar of a pickle or the meat which is salted, in that case the bacterial cell will be plasmolysed. That is, the entire content of the water will be drawn out and the cell gets killed. So definitely the meat or the pickle, they get protected or saved or conserved. Then weeds. What are weeds? They are wild plants which are unwanted in the agricultural tract of land. So they can be killed in playground by sprinkling excessive salts around their base. And again the answer is plasmolysis. And excessive fertilizers in the fields, they damage the roots and diminish the yield, because due to excessive fertilizers the medium in the soil molecules or the soil, it becomes hypertonic. This causes plasmolysis and this damages the roots, and naturally the yield is diminished. Now I am going to do a very important experiment today. There are many variations possible in this, children. We call this as Thistle funnel experiment, alright, or we also call it as an experiment to explain osmosis, right? Till now we have only discussed the definition of osmosis. Let us see the process in working. In your book, children, this is given at textbook page number here 38. By many modifications this has been given, like here, then explained all through, replaced by a whisking bag or a rubber sheet later, and then also muslin. So many hypothetical questions are possible. Let us begin with this one by one. Now, as I said, Thistle funnel experiment. What is a Thistle funnel? It is made up of glass and it is an apparatus like this. Open at both ends. You can see here it is open and here it is also open. But here we have tied a semi-permeable membrane. We take a beaker, fill it with water, and in a Thistle funnel, which is made up of glass, we add concentrated sugar solution. We fill it up to a certain level, let's say A, and then tie the mouth of the Thistle funnel by semi-permeable membrane. And we dip it by means of a stand, hold it in position into the water in beaker. And at the same time, this is the experimental setup. A parallel control will be set up, exactly the same, except that inside the Thistle funnel also it will be water. So that means water is outside, water is inside, and there is semi-permeable membrane in the Thistle funnel, again immersed in beaker. So let us see what is going to happen. First point, the level is marked in both the cases as A. Alright. Now what we see, after a few hours, the level of sugar solution in the Thistle funnel here is going to rise. Question arises, why? The question is why this level is rising. Again, children, keep in mind only the water molecules. Water is more outside and less inside. Why is it less? Because there's a concentrated sugar solution. That means more sugar and less water. So water molecules, they start moving through the semi-permeable membrane and they start entering the Thistle funnel. As a result, due to this, the level rises from A to B. Alright. Also, later it will be seen that the level in the beaker starts falling down a little. Why is it falling down? Because the water has passed into the Thistle funnel. Now, so many questions can be asked that what will happen if the water in the beaker is tasted? Will it be found sweet? No. The answer is just no, because there is no movement of sugar outside. The only movement that is possible in this setup is the movement of water molecules inside. I hope this is understood. Moving to the second one here. Now what will happen here? Water is outside and water is inside. So such kind of medium that is created is isotonic. So is this that it is possible here? Just not. There is no movement inside or outside, and thus this possibility is overruled and the level remains unchanged. So there is no movement of water molecules inside the funnel or outside from the funnel to the beaker, and also no drop in level or rise in level of the water inside the beaker. So this says clearly that level remains unchanged because there is no osmosis. Alright. So what do we observe from these experiments? Number one, that the semi-permeable membrane, it is allowing the water molecules. Suppose we have taken cellophane paper. So we can prove that yes, it is allowed molecules to pass through but not sugar, and thus it is semi-permeable. Right. Suppose we modify this experiment by slight changes in it. Instead of keeping sugar solution, concentrated sugar solution inside and water outside, see what we have done. we have kept more sugar concentrated solution inside, therefore I've colored it dark, and a very dilute or less concentrated sugar solution outside. Again, what will happen? Keep in mind only the water molecules. Again, in this case, if this is dilute, means there is more water and less sugar, and here it is less water and more sugar. So water moves from area of more to less, that is starts moving inside, and here the semi-permeable is already tied up at the mouth here. This is semi-permeable membrane. So the water starts moving inside and the level, just like this one, starts rising. So whether we keep concentrated sugar solution inside and water outside, water will rise, level in the Thistle funnel, or if we keep more concentrated sugar solution inside and less concentrated sugar solution outside, again the water level will rise. Now let's say a few modifications. If in the above experiment, instead of a cellophane paper or a semi-permeable membrane, right, suppose we use a rubber sheet. If a rubber sheet is used instead of a cellophane paper, suppose this was a cellophane paper tied up, but now we are using a rubber sheet, what will happen? Rubber, because it is impermeable, will not allow any water molecules from the beaker to cross over to the other side. That means in spite of difference in the concentration of water molecules, if we tie a rubber sheet here, naturally there will be no movement of water molecules. Reason? It is impermeable. Another question. If the membrane used is a muslin cloth is the one which has large amount of pores of mesh, or let's say a big amount of pores, and they're bigger in size. So what happens if we use a muslin cloth instead of a cellophane paper? The pores of muslin cloth are so large that they would not hold back even the sugar molecules, and the entire solution would flow down to a common level due to gravity. So muslin cloth is freely permeable for sugar solution. Suppose here there is highly concentrated sugar solution inside and less dilute concentrated sugar solution outside, and this is a muslin cloth. So that means water in that case should have moved inside, but because the pores of the cloth are so big that due to gravity the entire contents including water and sugar will be poured out into the beaker, right? That means they will be poured out into the beaker and the level of water of beaker will rise, but this will fall down to a common level. The level of Thistle funnel will fall down till it reaches a common level, and the reason is gravity. So this is the Thistle funnel experiment, children. As I said, in your textbook there are many modifications, and I have discussed all just now with you, right? Another modification is this, that what will happen, that if you're using a whisking bag like this, and same thing, concentrated sugar solution inside and outside is water, once again the answer will be just like this. So whether we use a Thistle funnel or we use a whisking bag, remember here we have already used a semi-permeable membrane, right? So that was cellophane paper. In this case, another semi-permeable membrane is whisking bag. So once again, the water molecules start rising because the water is more outside and less inside, and the level rises from initial to a final level, right? So I hope that is clear, children. This is a diagrammatic view of the movement of water molecules. The left side is depicting a dilute solution, right? Dilute means you can see the yellow molecules are water and the red are, let's say, sugar or salt molecules. So this is a highly concentrated solution, right? Because less of this is a dilute solution because there are more water molecules and less of sugar molecules. Whereas on the right side, this is a highly concentrated solution now because there are more sugar molecules and less of water molecules. What divides them is a semi-permeable membrane which allows only the water molecule. So see the arrows, they are only moving from left to right. So that means sugar is not moving at all. What is moving is water from a region of more to region of less, and it will continue to move till an equilibrium is obtained. And that will be our last thing for today, that how long can osmosis continue? That means if I ask you in this question that, alright, this has been done like this, but how long should osmosis continue? And the answer is that theoretically the water molecules will start rising, right? But practically the answer is something else. Let's see what. Theoretically, osmosis should continue until the concentration of water molecules becomes equal, that is isotonic, on both sides of the membrane. But practically, the increase in height of rising column reduces further osmosis. That means the water molecules theoretically should continue to rise till an equilibrium, that is, water is equal inside the Thistle funnel and also outside in the beaker. But practically this is not obtained. Reason that this level of height that is increasing and the weight of this water will prevent the further rise of water molecules. So that you'll have to explain that theoretically it should continue till equilibrium, but practically that is not achieved because of the weight of this water column that prevents the further entry of molecules. So this we are going to stop today, children, and my next topic would be uses of turgidity to plants next time. That will continue from here. So kindly go through these pages, see that you are able to understand, children. All these I have covered. Page number 40 and the next page, that is 41, also we have studied. So my next topic, even this we have discussed last time, children. Different types of pressure, wall pressure, turgor pressure, and these processes, the flaccidity, turgidity, and plasmolysis, deplasmolysis. So here I'll start from next time, uses of turgidity, and there are 3 points under this that we'll start next time. So children, for today I'll be sending you the pages to write. Kindly make a note of the same. Very less work for today to write, children. Only the paragraph here about tonicity and 3 types of solutions, and over here one paragraph about semi-permeable membrane and plasmolysis in practice, and yes, the Thistle funnel experiment. It's more important that you understand the same and certain questions that I have framed hypothetically, but yes, they're very, very important. So that's all for just now, children. Any questions, please keep posting and write the notes regularly. Thank you.